The next category of tests that we're going to talk about that can be used to diagnose infectious disease are immunologic methods. We can also call these serologic methods. This is going to include anything that's going to utilize the power of antibody-antigen interaction. And so things like agglutination, precipitation reactions, we can do fluorescent tags on antibodies, we can do lateral flow assays or immunochromatography work, as well as ELISAs. And so we'll look at each one of these in a little bit more detail throughout this lecture. It's important to start out this discussion with defining two very important words when we're talking about immunologic testing, and that is specificity and sensitivity. So when we're looking at serologic tests or immunologic tests, there's going to be a degree of specificity of each test, and that is defined as the property of a test to only identify a specific or certain antibody or antigen, and then disregard all other proteins that might be around. So when we say a test has high specificity, that means that it is able to detect a very specific antigen or antibody, depending on which one we're looking for, and then not pick up anything that isn't that specific antigen or antibody. But then there's also the term sensitivity, and that is a reflection of how small of a quantity needs to be present in order for the test to be able to find it. And so it reflects the degree to which a test will detect that antigen or antibody in every positive person. So a test that has high sensitivity is able to find or identify or become positive or indicate positivity even when there's a very, very small amount of antigen or antibody present. So then the goal of any immunologic test is to have high specificity and high sensitivity. Sometimes tests are going to have either one or the other, and maybe they'll have really high sensitivity, but it's not very specific. And so you could get some false positives. Or you could have high specificity and low sensitivity, and therefore you might get some false negatives. So the sensitivity and specificity measurements are very important when we're looking at immunologic testing, and most often these tests are going to have, like the kits that these tests are part of, will have a sensitivity and specificity rating on them. You've probably picked up that I've used the term serologic testing with immunologic testing, and I've kind of been using them interchangeably. That's because when we're talking about serologic testing, that is anything that's going to be utilizing an antibody to recognize an antigen, which is the premise behind immunologic testing. So serologic testing is in vitro testing, so not inside of the body, but in a test tube or on a test cartridge or in a 96-well plate or whatever, just not inside the body, but testing plasma or serum, urine, cerebrospinal fluid, really any of these body fluids for the presence of specific antibodies. The idea behind this, or it could be antigen as well, the idea behind this is that antibodies are produced by the human immune system, and they're proteins that have an ultra-specific antigen that they recognize. So we can create tests that have this antigen in it, and we can look for antibodies that can bind to that antigen. And if that is the case, we can visualize this by attaching something to that antigen. We can attach an enzyme, we can attach a fluorescent protein, we can attach a piece of latex, whatever it might be. We can also do the flip side and have antibody in our test and look for a specific antigen. And then we can tag that antibody with an enzyme, with a protein that fluoresces. We can also attach that antibody to the surface of a well plate or to the surface of some cellulose paper or something like that, where we can create tests that are going to look for the ability or look for any antigen antibody interaction. And this can be used to determine the immunologic status of an individual. So if a person has an antibody to a certain antigen or to a certain pathogen, we know that individual has been exposed at some point in their life. We can also use this to diagnose if a person has a certain pathogen, and we can use it as a quick screening device for individuals to see if they have a certain disease or not. So the power that we have with serologic testing is really pretty awesome, and we've been able to create a lot of rapid, so fast, but also highly sensitive and highly specific tests using serological or immunological methods. Let's see what this looks like in diagram form. Now this picture is actually split into two separate situations, question 1 and question 2. Question 1 is asking, does the patient make antibodies towards a certain microbe? And that means, has the patient been exposed to this pathogen or to this microbe and mounted an immune response against it and would have circulating antibody in the plasma or in the serum? So what we can do is we can take a known antigen and we can tag it with, in this case, we're tagging it with a fluorescent protein, and we're mixing patient plasma or serum with a known antigen tagged with fluorescence. And then we're looking, or it could be a piece of latex as well, and we're looking to see if there is agglutination or attachment of this antibody to antigen. If the antibody is there, then the patient has been exposed to antigen and has mounted an immune response. Now, the second question on the other side is patient sick. Here is the microbe that we've grown out of the sample from the patient. What is it? So what is the identity? Well, what we can do is we can have some suspect identities, and we can, as humans, have developed antibodies that are known to bind with certain antigens. And so we have an anti-serum here that recognizes Staph aureus. If we take a colony that we think, this patient might have Staph aureus, and we mix it with antibody against Staph aureus, and if we see clumping or agglutination, because there's going to be little beads attached to those antibodies, then we know that that identification of that sample or of that pathogen or microbe is Staph aureus. If we don't see any clumping, then we can say, well, it's not Staph aureus, it must be something else. And these are ways that we can use these powers of antibody interaction to answer questions about patients' diagnoses. Agglutination and precipitation reactions are named so because what we're looking for is agglutination or clumping of antigen and antibodies together and then coming out of solution. So that's where the agglutination and then precipitation, right? The falling out of solution. And what these kits look like is there are antibodies that will have a latex bead attached to them, or you can have other particles attached, usually a latex bead. You can also utilize just like red blood cells as well. There's a variety of different things that we can visualize clumping, but it's really anything that's going to be able to be visualized when you get enough of these particles together. And what we see happen is when there is an antibody interaction during an agglutination assay, you have multiple antigens brought together with multiple antibodies interlinking, and that will cause for clumping to occur and for those antibodies to fall out of solution or precipitate. And we're able to visualize this as clumps. And so the picture I have shown here, we're looking at a strep typing kit. And so we have group A strep and we have group B strep antibodies, and we're mixing them with different bacteria. So the antigen is on the bacteria, and if the antibodies recognize it, then they'll be clumping. And so in the first circle, you can see that it's pretty uniform, pretty clear. It's blue, but it's not clumped together. Like we see in the right-hand circle, we actually can see some visible clumps. And so the visible clumps show us a positive antibody-antigen interaction, which would indicate that that test would be positive for whatever antigen we're looking for. Immunochromatography is kind of a big word, but if you break it down, it starts out with immuno, which you should start to think, okay, it must have something to do with the immune system. And then chromatography. Chrom is color, graph is your record, right? And when we talk about chromatography, we're talking about some sort of visual record that we can see. So with immunochromatography, we're looking for a visual record that can explain a result to us. Either say, yes, something is present, or no, it is not present. So one of the forms of immunochromatography is a lateral flow assay. This means that it's going to be lateral, so flat on usually like a piece of cellulose paper or filter paper or something like that. And this is pretty much what we see like in a pregnancy test or a rapid strep kit. They are going to utilize lateral flow assays. Rapid COVID testing is a lateral flow assay. And there's this cartridge or a cardstock card that has a porous material, almost like a thick filter paper. Can be cellulose, it can be a polymer of other materials, but whatever it is, it has the properties that liquid will flow through it by capillary action. And what happens is a sample is put on one end of the cartridge and it travels across laterally to the other end of the cartridge, just by, like if you put a piece of paper towel in water and how it moves through like by capillary action. That's the exact principle then behind a lateral flow assay. The difference here is that with a lateral flow assay, there are going to be different spots on that paper or on that cartridge that the fluid is going to pass through. And along those spots, there's going to be antibodies that it can interact with, that the sample can interact with. And then there's multiple steps. And with each step, there's going to be a different set of antibodies that might have a little bit different specificity. So I think the easiest way to do this is by looking at a diagram that shows a lateral flow assay with all the steps. So I'll do that in the next slide. Here's a diagram of a generic lateral flow assay. And it's just one strip, but each strip is showing you a different step in the process. So imagine that the analyte or the antigen that you are looking for, and let's just use COVID-19 as an example. So when somebody is suspect of having COVID-19, they'll go in and they'll have a rapid COVID test done. And so there's going to be a sample, either nasopharyngeal or a nasal swab, and it's going to be mixed with a solution so that it's going to be suspended in this liquid. That is called the analyte. The analyte is going to be that antigen that we're hopefully going to find. Now that drop is placed on one end of the paper, and it's going to move through by capillary action. The first thing it's going to come across is a strip that's going to have antibody that recognizes the antigen, or antibody that recognizes COVID, or the SARS-CoV-2 piece of the virus, and it's going to pick up those antibodies and drag them along with the fluid as it moves through. So now we're on the second layer, and then the second image, right? Okay. And then there's going to be another line of antibodies that are also going to recognize that same antigen, SARS virus, and bind as well. And so now we have an antibody sandwich containing the analyte in the middle. And those antibodies that picked up the analyte on the second time are embedded into the cellulose, and so they don't move. And then those first antibodies had some sort of substrate or some color on them attached to it. But not all antibodies are going to bind, and so the ones that don't bind are going to continue on through and hit the control line. And that is why we need to have two lines that are visible in order to call a test positive. And then those yellow things are the color or the whatever it is, latex, a fluorochrome, it's not going to be fluorescent, but a substrate, some sort of enzyme, whatever it might be to produce a color. And then we can visualize those lines. And so then if you have one line, the control line only, then the test is negative. But if you have two lines, you have the test line and the control line, then that test is positive. I've mentioned a few times that fluorescence can be used when using immunologic methods. And so let's quickly look at the process of immunofluorescence. So using fluorescence in immune testing. And this is going to utilize what are known as fluorescent antibodies, or FABs. And what that means, it's an antibody that is made for a certain specificity, or a monoclonal antibody, that has a fluorescent dye attached to it. So in the lab, we can make monoclonal antibodies for whatever antigen we want by engineering a process to develop these antibodies that have one single specificity. That's where monoclonal comes in. We throw a fluorescent tag on the back of that antibody, and then we can mix that antibody with any sample we want. And if there is attachment to the sample, then that means its specific antigen is present. And this is the direct testing method of immunofluorescence, where a sample is taken from a patient where we don't know, or it could be from an organism, and we don't know if a certain protein is there or not. We add monoclonal antibody that is specific for that antigen. Then we do a few washing, it's like staining with these monoclonal antibodies, a few washes, and then we look at it at the microscope, and if we see fluorescence, that means that antibody has bound to the surface of whatever we're looking at, and that means that that specific protein is present. And so it's very valuable to determine if a pathogen is present. And we can also be able to determine exactly where in a sample a pathogen is either living or what it's bound to. And so using immunofluorescence is a pretty great way to not only identify a pathogen, but also find out a little bit more about its location and even amount in a sample. Immunofluorescence testing can also be done in an indirect manner as well. We're still utilizing monoclonal antibodies that have fluorescent tags attached to them. It's just this time we're utilizing two sets of antibodies. One is going to be bound to the surface of the pathogen, and then the second antibody that actually has the fluorescent tag is going to recognize antibody. So you can see from the picture here, the top pathway is direct fluorescence, where we have the fluorescently tagged antibodies attached directly to the pathogen or to the antigen. Where with indirect, we have an extra step in there, where we either add antibody that would recognize the pathogen that doesn't have fluorescence, or there's already antibody attached to a pathogen that is of natural origin, so there's no fluorescence. And then we add a monoclonal antibody that has fluorescence attached to it, but it's going to recognize antibody. And then indirectly, it's showing you, yes, the antigen is there because that antibody is recognizing antibody that is recognizing antigen. So that's where the indirect part comes in. Enzyme-linked immunosorbent assays, also called ELISAs, are another way that we can use antibody-antigen interactions to look for the presence of either an antibody or an antigen. Depends on what we're looking for and how the test is developed. Overall, ELISAs are going to utilize enzymes that are linked to antibodies, and then a substrate for that enzyme is added, which if the enzyme and substrate interact, then there's usually going to be a color change. And then that color change indicates the presence of a certain antigen or antibody, depends on what we're looking at. And ELISAs require a solid surface to interact with. And so usually this is going to be done in a 96 well microplate. Because of the structure of that microplate, that plastic surface is able to take antibodies and have them adsorbed to the surface of that plate, or antigen can be adsorbed to the surface of that plate to allow for interaction to occur. So we'll look at two different ways that we can use ELISAs in the next couple slides. With ELISAs, we can have both indirect or direct testing methods. The picture here shows what an indirect ELISA looks like. This is in your text, so definitely take time to go through and look at it a little bit closer in depth, but it's showing you both positive and negative side by side. So well A is going to be the positive ELISA, where well B is the negative ELISA. What happens with indirect is that the antigen of interest is going to be adsorbed to the walls of the 96 well plate, and then the patient's sera is going to be mixed in those wells, and this test is going to detect if a patient makes a certain antibody. So if the antibody is present, it will bind to the antigen that's stuck to the surface of the well. If it's not present, nothing's going to bind. And then there's several washes where there's going to be a buffer added and then rinsed out to remove any antibody that isn't bound. Then there's going to be an antibody that has an enzyme attached to it that is going to be added that will recognize any bound antibody and make an antibody sandwich, really. So you have antigen, antibody, and then antibody that recognizes antibody. If the antibody isn't there, then none of that enzyme-linked antibody is going to stick, and then you wash it. And then you add the substrate, and then if the enzyme is present and the substrate is present, then there will be the enzymatic reaction, which will result in a color change. And so the color change is only going to occur if the patient has antibody to that specific antigen that was part of the ELISA. And so we call this the indirect method because we're using two different antibodies, because we're looking for that specific antibody binding to an antigen. With a direct ELISA, this is going to be very similar to what we saw with the indirects, but this time we are looking for antigen in a patient sample. So antibody that is specific for that antigen is going to be adsorbed to the sides of the well or the bottoms of the well, and then there will be patient sample mixed with the antibody, and if the antigen is present, it will bind. Then there'll be washing to remove any unbound antigen, and there will be an antibody that has the substrate or the enzyme attached, and then it will also recognize that antigen, and so you end up with an antigen sandwich where you have antibody, antigen, and then antibody that has the enzyme on it, substrate is added, and then a color change will develop if that antibody that has the enzyme is attached. And so we have a positive direct ELISA when there's a color change. So very similar to the indirect. It's just with indirect, we have the antibody or the antigen bound, to the wells, and with a direct, we have the antibody bound to the wells. Included in our list of immunologic methods, we have something called antibody titers. We can find specific antibody in patient plasma or serum, and we can measure how much antibody they have in a certain sample, and we call this a titer. And this is done by serial diluting patient's serum or plasma and looking for the highest dilution or the smallest concentration of patient sample that is still able to produce an agglutination reaction with a certain antigen. What this does is it tells us potentially how strong an individual's antibody response would be to any type of infection. We have run with COVID-19 antibody titers and followed out how many months after infection a patient has circulating antibody. Over time, that decreases, and that can all be measured by doing antibody titers. The last group of serological testing that we'll talk about is serotyping. With bacteria, we have another breakdown past a genus species called a serotype. So Salmonella, for example, Salmonella enterica is a species, but it has like over 2,000 different serotypes, and it all has to do with the protein profile on their surface. It's important to know different serotypes because some of them are going to be pathogenic, some of them will be pathogenic, and then we can also link people together in an outbreak based on which serotype we're seeing in a population. This is going to use antibody antigen interaction, just like all other immunologic methods. And this time, the antibodies are going to be in the test kit, and somehow they'll be like an agglutination or some fluorescence or something, just like we've seen in other types of tests, to determine what serotype different bacteria are.